All right, hello everyone, and thank you for coming to our November meeting. Just a couple of announcements to get us started. The Myco Talks is next week, next Thursday, and I'll put the information into the chat on how to register to that. For that, the speakers are David Denning and Anita Sill, so it should be two fantastic talks. And as always, please enter your questions into the chat, and we will take all questions after the speakers. I will turn it over to Lillian, who will introduce our first speaker for the day.
Thank you, Jessica. The first speaker today is Allison Porman Swain, or Allison Porman if you are looking for her papers. Allison completed her undergrad degree in molecular biology at Lehigh University. She then moved to Brown University for her PhD, where she joined Richard Bennett's lab. And then she's now a postdoc in Aaron Johnson lab at the University of Colorado Medical Center. And Allison is currently on the job market. So if you know any position, get in touch with her. Allison, we are waiting to hear you talk.
Yeah, thanks so much for having me today. This is the first time I'm really presenting this work. It's unpublished work. So thank you all for being here. So my main focus for research is Candida species. And these are usually commensal organisms, so they can live on us without causing us much harm, but they can cause these opportunistic infections in individuals that are immunocompromised, such as people with AIDS or people undergoing immunosuppressive drug treatment because of an organ transplant. And these can result in mucosal infections, such as oral thrush, other mucosal parts of the body, and they can ultimately lead to these systemic bloodstream infections. And as you all probably know, as eukaryotic fungal infections, they're difficult to treat. And once Candida gets in your bloodstream, it's really not a great prognosis. These systemic infections have about a 50% mortality rate. So it's pretty bad prognosis if Candida gets in your bloodstream. And Candida albicans is the species that really causes, is the most common cause of these Candida infections, and it makes up about 80 to 95% of these infections clinically. And so one feature of Candida albicans that's really thought to promote its ability to colonize and infect humans is its phenotypic flexibility. And so it has the ability to kind of switch between all these different phenotypes. And some phenotypes have been identified, such as this gut phenotype that enables colonization of the GI tract, as well as the yeast type transition, which is known to be necessary for virulence and kind of tissue penetration by these cells. So all of this ability to kind of switch between these phenotypes regulates the commensalism and virulence of Candida. And I study this specific phenotypic switch known as the White-Opaque switch. And so this is a bistable switch between these two phenotypic states. White cells are kind of in this round budding yeast state, whereas opaque cells appear more elongated. They have kind of darker colonies on plates. And this is an epigenetic switch. So they're really just changing the genes that they express. There's not really any underlying genetic changes. It's heritable, meaning once they switch into one state or another, they can proliferate in this state. And it's also reversible. So certain cues are known to cause the yeast to really switch back and forth between these states. It regulates the differential expression of around 1,000 genes, and it's known to be affected by the environment. So different signals will cause the cells to switch in one direction or the other. And this switching regulates niche specificity, so where the cells prefer to occupy the host, interaction with host immune cells, how they respond to the environment, as well as their metabolic preferences. And a key aspect of Candida albicans biology that this switch regulates is their mating. And so Candida albicans is kind of this weird, it's called the parasexual cycle because it's not really a complete meiotic mating cycle. And so kind of the first step in this mating process is that actually cells must switch from this white state. This white state's really not able to undergo very efficient mating. So it's considered the chaste state. And so they have to switch to this kind of switch to the opaque state in order to actually undergo efficient mating. And then once they do mate, actually genes at the mating type locus in these A alpha cells will cause them to switch back to the white phenotype. And A alpha cells really kind of prefer this white phenotype. And so it's only really A and alpha cells that undergo this white opaque switch. And then these tetraploids actually undergo this weird kind of concerted chromosome loss process to return to the diploid state. And so that's why this is kind of the parasexual cycle. There's no meiosis that's been identified in Candida albicans. And so in my graduate work, I'll briefly tell you what I was looking at. And so when I started, really the white opaque switch had only been identified in Candida albicans and its sister species, Candida dubliniensis. And so my question was, can this closely related species that's also a pathogen, can this species undergo white opaque switching and mating? This species was believed to be asexual and mating had never been observed before. So I thought maybe if we could identify kind of this white opaque switch, we could see that they mated. And so yes, that's what I found. So I identified this white opaque switch in tropicalis that acts to similarly regulate mating. So the opaque state mated much more efficiently. And yeah, it was the first time sexual mating was identified in this species. And so I also did some gene expression analysis with a microarray back in the day. And so we kind of made these custom tropicalis microarrays, and we identified about 400 genes that were differentially regulated, including the master regulators of the switch, which seemed to have conserved functions. And so this was a really exciting project. I went on to find some cool differences between tropicalis and albicans, but I won't go into those details now. So I'll just to summarize the background, Candida species are these human fungal pathogens. The white opaque switch in Candida albicans regulates their ability to mate. And my graduate work uncovered white opaque switching and mating in Candida tropicalis. So now going into kind of what my postdoctoral studies have been focused on is this RNA modification that's called N6-methyladenosine or M6A. And so it's this methyl group that gets added to adenosines within RNA. There have been known readers identified. So in mammalians, these are the YTH proteins. In yeast, they have a conserved reader called the methylated RNA binding protein or MRB1. And writers have been identified in mammalians. These are metal-314 complex. And in yeast, this is the IME4 gene. And I'll say that these proteins are pretty well conserved. They really have key amino acids that are conserved from yeast to humans. There are also known erasers in mammalians that have been identified, but none have yet to be identified in yeast. So we're not sure if there really are any erasers or we're just still looking for it. So yeah, this system's pretty well conserved, and M6A has been shown to play roles in RNA stability, typically destabilizing its target RNAs. It can play a role in messenger RNA translation, where it typically enhances the translation of a message that it gets modified. And it's also been shown to play roles in RNA localization. And I'll say that a kind of a theme in the M6A field is that its function is often context specific. So in different cell types and different even messages that it modifies, its function can be very specific for that context. And so my postdoc work in Aaron Johnson's lab has really focused on looking at this RNA modification in breast cancer cells. And I found this pretty cool function for it on this long non-coding RNA, hot air. And so this work is up on bioRxiv, hopefully to be published soon. You want to check it out. But for today's talk, I'm really going to focus on my path to kind of studying M6A in Candida albicans. And so, you know, kind of combining my postdoc work in breast cancer, looking at M6A, and my background in Candida biology, I asked the question, what is M6A doing in Candida albicans? And so we know kind of the function of IME4 based on work done in Saccharomyces cerevisiae. And so just to briefly introduce this, I remember I told you IME4 is this M6A methyltransferase. And IME4, inducer of meiosis, it plays a role in the induction of this meiosis in A alpha cells. And so IME4 will lead to the induction of IME1. And actually my collaborator recently identified a specific M6A site in the 3' UTR of RME1. And so this IME4 basically acts to modify RME1, ultimately causing its degradation, and that allows IME1 expression to get turned on to then promote my entry into meiosis. And another cool aspect of IME4 in cerevisiae is that it is regulated by this antisense transcription. And so in this case, in haploid cells, you have transcription in the antisex direction of this RME2 non-coding RNA. And this transcription event really prevents expression of IME4 in this context, where cells do not want to enter meiosis. And then in A alpha cells, the A1 alpha 2 complex that's formed by genes at the mating type locus will actually block expression of RME2 to allow for IME4 to get turned on and upregulate this M6A during meiosis. And so, you know, my question was, what is IME4 doing in Candida albicans? The species doesn't have meiosis. Has anyone looked at this or studied this? And so I turned to PubMed, did a quick PubMed search on IME4 and Candida albicans, and no results were found. So I was like, okay, maybe no one's looked at this, but I didn't stop there. I turned to Google and I did a similar search. And actually what I found was on this Grantome website, a former F32 fellowship that was looking at exactly what I wanted to look at. So IME4 in Candida albicans and its role in mRNA methylation. And so this was a postdoc fellowship from Guy Bushkin when he was a postdoc in Jerry Fink's lab at the Whitehead Institute. So I ended up reaching out to him, and he had really shifted gears into working on the Saccharomyces cerevisiae stuff. So he was really happy that I was interested in kind of picking up where he left off on this project. And so he shared with me some of his data in the function of IME4. So he created these IME4 mutants that had a deletion of IME4. And the first thing he noticed just by kind of growing these cells at room temperature is that wild type cells, when he grew up an opaque colony and plated it to media, he saw kind of a mixture of white and opaque colonies that would form. So the opaque state isn't super stable, I'll say, at least in this specific strain of yeast. And so white colonies kind of appear a bit lighter color here, whereas opaque colonies are darker because they absorb this dye, floxin B. And so he noticed that the IME4 mutants actually the opaque seem to be a bit more stable, and he would really get these pure opaque populations. So another feature of the white opaque switch is that this high temperature, 30 to 37 degrees, will really cause opaque cells to switch back to the white state pretty efficiently. And so when he grew cells at 30 degrees, he noticed that the wild type cells all were kind of switched back to this white phenotypic state. And we can see that at the cell level here, where these are really round, the round kind of budding yeast that we typically think of. While the IME4 mutants were able to stay in opaque even at this kind of higher elevated temperature. You can see that the cells really remained elongated. And so it seemed like these knockouts, these IME4 knockouts really had a stabilized opaque state. He also looked at mating. So he performed these mating assays where he could plate these lawns of MTL alpha cells that had a specific auxotrophy, and then spot MTLA cells that had a different auxotrophy. And so he could spot these wild type or IME4 knockout cells, and then plate these to selective media so that only the mating products are able to grow, and kind of the number of mating products would indicate how efficient mating was in this context. And so what he found was that in wild type cells, while they underwent some mating, the IME4 knockouts had a lot more efficient mating than wild type. And so it seemed like these IME4 mutants really had an increase in this mating. And so what we think is that IME4 is really playing a role in this process of switching opaque cells back to the white state. And it also makes sense that kind of these knockouts have more efficient mating because we know the opaque state is this mating competent state. So Guy was nice enough to mail his yeast strains to me so that I could kind of pick up and do some RNA sequencing analysis to determine kind of these, what gene expression differences are there between wild type and IME4 knockouts. And so we first looked in opaque cells. And in this context, we identified quite a few genes, actually 466, that were differentially expressed between wild type and IME4 knockout cells. Genes that were down in IME4 knockouts were involved in cellular response to stress and regulation of transcription. Genes that were up in IME4 knockouts were often involved in metabolism or mating, and I've listed some specific genes of interest down here. Now, we also looked in white cells, and here we did not see many genes that were differentially expressed, possibly suggesting that, the white state is able to kind of still be consistent even in the IME4 knockouts. Although the genes that were differentially expressed were enriched for genes involved in cell-cell adhesion, flocculation, and filamentation. And some genes are listed down here. So this was, good to see, but we wanted to really map the M6A sites to identify what genes are actually getting modified. And so to do this, I actually was a recipient of one of these RNA epigenetics grant awards from Eclipse Bioinnovations, and I was awarded one of these free M6A eClip kits, which is really nice because all of my funding was for breast cancer research, and so it really helped me kind of get started in mapping M6As in the yeast cells. Not a super cheap experiment because you need a lot of antibody, you need a ton of RNA, and but yeah, thanks to Eclipse for this. So in white cells, we identified about 193 M6A sites, and there were M6A sites present in some white-opaque regulators, as well as genes involved in RNA metabolism and response to chemical or cellular stress. Some genes of interest listed down here. And in opaque cells, we identified 136 M6A sites. These were in genes involved in symbiotic interaction, metabolism, as well as a pretty good amount of heat shock proteins. So this could be possibly one of the ways that opaque cells actually respond to that elevated temperature to switch back to white, is maybe through M6A modification of these heat shock proteins. And so we, of course, wanted to compare, our M6A modified genes and our differentially expressed genes. And we found a pretty small overlap between the two. So out of our 138 M6A sites and 466 differentially expressed genes in opaque cells, only 12 were shared between the two. And of course, the situation was even less in white cells. Well, we only had identified 18 differentially expressed genes, but only 2 genes were really kind of shared this M6A and differentially expressed pattern. And so what this really suggested is kind of either A, we have maybe a lack of detection of some M6A sites. For example, if M6A is really efficient at destabilizing its targets, maybe we're not actually detecting all the M6A and they're just getting degraded really quickly. The other possibility is that M6A and Candida albicans might act to either regulate translation, so we might not see changes at the RNA level, or potentially localization as to a greater extent than RNA stability. And so these are, 2 possibilities we kind of want to look into further in the future. So another interesting thing that we noticed from this M6A mapping is that we found some genes with alternative M6A sites in white and opaque cells. And these were often in morphology or adhesin related genes. And so of our 18 kind of common M6A genes in white and opaque cells, there were actually 12 that showed this differential M6A pattern. And only 6 M6A sites were shared between white and opaque cells. So this could be something to look into in the future, is maybe this is a way of differentially regulating these genes in white versus opaque cells. So some other interesting takeaways from mapping these M6A sites, we noticed that most of these sites were detected in coding sequences. So only about 17% were detected in 5' UTRs and even less in 3' UTRs. So this, I think, suggests a potential for regulating translation, especially, it's in these messenger RNA coding sequences. So it could be regulating translation efficiency there. We also found M6A sites in intron-containing genes, as well as some major repeat sequences and retrotransposons. And so this could possibly suggest a conserved role in regulating splicing and expression of these kind of repeat sequences, where it's known to, in mammalian cells, it can repress expression of these repeat sequences. So just to summarize on kind of the progress we've made so far, we've found that Candida albicans IME4 regulates white opaque switching and mating. And we think it's really playing a role in induction of this opaque to white switching. We've identified hundreds of genes that are differentially expressed in IME4 mutants and hundreds of M6A modification sites. So in the future, what I really want to look at is, what are really the key M6A sites and kind of the functional outcomes of this M6A modification in those genes, and how does that act to really regulate this white opaque switching and mating. Secondly, I'm really interested in how IME4 expression, as well as induction of the M6A modification, might respond to the environmental signals, and how maybe we know that there are these metabolic differences between white and opaque cells. Does IME4 help mediate these changes in metabolism? How does IME4 regulate interactions with the mammalian host? So we've identified several differentially expressed genes and M6A modified genes that are known to play a role in interactions with the host. So we think that these IME4 mutants might have altered commensalism or virulence in the host. So that's something I want to look into. And finally, is this function of IME4 and M6A conserved in other Candida species? For example, in Candida tropicalis, another species that we know has the white opaque switch, does IME4 have a similar role in that species? And what about other related species that don't have white opaque switching or meiosis? So that's something I kind of want to look into in the long-term future. So just to thank my postdoc advisor, Aaron Johnson, Justin Roberts, who's a graduate student in the lab, who's been a huge help with all of the bioinformatics analyses. My collaborator, Guy Bushkin, who really started up this project, my PhD advisor, Richard Bennett, and Jerry Fink for helping Guy start up this project in his lab, as well as my undergrad mentee, Hamza Ahmed, who's really taken an interest in working on this project. We've had fun over the summer, and he's continuing on in the lab, so it's exciting. Also, thanks for my funding. Fingers crossed for my K99 application that's currently under consideration on this work. I really hope I can get some funding to follow up more on this work. And I'm also currently seeking faculty positions. So if you know of anything or have any insight or advice, I'm happy to take it. All right, with that, I will pass it over to Axel.
Yeah, we have so far 2 questions for Allison. So the first one is from Jessica. And Jessica say, I'm very intrigued by the difference in the number of IME4 regulated differentially expressed gene versus the number of M6A site, particularly in white cells. Would you comment further on the potential biological differences between these 2 gene sets? Do you think that the IME4 might have multiple function?
Great question. So yeah, in white cells, there were only like 18 differentially expressed genes, but there are almost like 200 M6A sites that we identified. So yeah, one possibility is that, white cells we think that they're kind of less different between the wild type and IME4 knockouts, because it seems like they're able to be stable. They're able to kind of exist in that white phenotype. And the conditions that I grow them in, they do seem to be a pretty stable phenotype. But yeah, I think they're, why are we detecting M6A sites here? Like, are they doing anything? What are they doing? I think that's still definitely an open question. And so one thing we want to look into is the role of the reader protein in yeast, the MRB1 gene. And, we haven't really examined if this gene is regulated by the white-opaque switch, and also what it might be doing to affect these M6A modified messages. And so, we think that potentially like that could be a way, either through differential expression or maybe differential function, because we do know, there are about 1,000 genes differentially expressed between white and opaque cells. So there could be some sort of effector different between the two that's causing the kind of this differential regulation of M6A. I think there is another question of, are there M6A independent functions of IME4? And yeah, I think that's definitely an open question. And we do actually have, one thing that Guy helped develop in Jerry Fink's lab was getting CRISPR up and running in Candida albicans. And so he did create an IME4 mutant that only has the active site mutated. And so I think that strain is maybe something we can use. It does have a similar phenotype where opaque cells are stabilized, but it also, I think, might have some differences between the total knockout as well. So I think that's kind of a strain that we might be able to use to get at that question, does IME4 have any M6A independent functions? So stay tuned. I think that's definitely still a question of like, the differences between this M6A output and versus opaque cells.
Yeah, and the second question is from Mark, and he's asking, under what condition did you check the differentially expressed gene in the IME4 mutant? Were these hyphae inducing condition or yeast state? And then he's asking, since some of the genes that you identified in the opaque cell are normally high induced in hyphae, and involved in evading immune-induced stress?
Great question. So yeah, I grew these cells in YPD, and I grew them at just room temperature because opaque cells, will switch at any higher temperatures. So actually I grew them at like 22 degrees Celsius, and it was kind of my best way to really keep or try to keep the opaques opaque. Even under these conditions, opaque cells aren't, the wild type opaque cells at least, aren't super stable. So the other thing about the white-opaque is that glucose itself is actually a signal for cells to switch to white. And so being grown in kind of that rich glucose condition, opaque cells kind of will often switch back to white, and I really have to monitor them closely to make sure I'm actually still growing opaque cells. So that's one of the challenges. And so, that I would say opaque cells aren't like super happy in these conditions. They're definitely still in yeast, so it's not inducing hyphae, but they may be a little bit stressed, I would say, like in that glucose. They just metabolically do not prefer to have glucose around. So your other question was, related to the SOD5 gene. So this is one of the genes that was upregulated in opaque IME4 knockout cells. And it's also one of the genes that we identified to be M6A modified in opaque cells. So another thing about SOD5, it is induced in this hyphae conditions. It can also be induced in neutrophils. It's also can be induced by osmotic and oxidative stress. And so we know from human cells actually that M6A plays an important role in hypoxia. So M6A can be upregulated, and there can be kind of this increase in M6A modification of messenger RNAs under these hypoxic conditions. And so I think maybe that is possibly one of the reasons that we're seeing this regulation of SOD5 in our opaque cells. Maybe actually, since these cells are a little bit stressed, they're starting to upregulate some of these stress genes, and these might actually be regulated by IME4. So I think that will be an interesting thing to look into of, how is IME4 expression and M6A, overall M6A modification regulated by these different environmental conditions. And so I think, one context where I really would like to go back to and look at gene expression and mapping M6A sites is the condition where we know IME4 is playing a role. So when opaque cells are grown at these higher temperatures and they're switching back to white, I think maybe that's actually someplace where M6A could actually be induced and upregulated to help mediate that switch. So that's something I have on my to-do list.
Allison, there is a follow-up from Mark. Yeah, I actually meant the condition for the white cells. So if you grow these as yeast, you won't see the effect of IME4 on SOD5 because it's not that much expressed in yeast.
Yeah, that's a good point. So yeah, we didn't see differential expression of SOD5 in these IME4 mutants in white cells. But yeah, Guy did look a lot he looked extensively for different phenotypes of these IME4 mutants. And he did look at hyphae and didn't see a major difference there. But, I think the white opaque switch regulates hyphal formation in a lot of different ways as well. So I think that is something I ultimately also want to look into is, does IME4 have an effect on hyphal formation, biofilm formation, and things like that? Because, I think, it has potential to have a lot of different effects. But yeah, good question. I think I still definitely need to do some more analyses on what IME4 might be doing in these other different contexts.
All right, thank you very much. If anyone has further questions for either of our speakers, feel free to turn on your mic. Otherwise, thank you both for fantastic talks, and thanks everyone for coming. And we'll see you in December.
Thank you so much for having us.
Thank you very much.